The native plants of Texas are the only plants for me.
Andi attended Texas A&M University, where she studied agriculture, microbiology, and bioremediation in the environmental sciences program. Andi's a soil health practitioner, a science communicator on a mission to help others restore life and functionality to their soil systems. She is thoughtful about the interconnected nature we have as stewards of our landscapes, and wakes every day inspired by the resilience and willingness of individuals to develop meaningful relationships with soils, plants, and greater environment on which we depend. All right, Andi, I think you're ready to share.
So yes, today I'm going to be talking with y'all about some of the things that I see on a microscope and how we can use those insights in native landscaping. Y'all did such a great job introducing me. I'm really grateful to be here. I've had a chance to work with some Texas master naturalists here in the Austin area for an urban forest site called Circle Acres. And I've just been very encouraged by the kind of devoted volunteers that we have through that program. I also grew up in North Texas. So I grew up in Grapevine, Colleyville area, and Fort Worth Botanic Gardens, I understand, is where you're meeting today. That's actually where my brother proposed to my now sister-in-law. So there's lots of roots there. And I just wanted to mention that I still get back there every now and then. So when I just to expand on my background a little bit, when I first got interested in soil microbiology, it was during my time at A&M, and I ended up taking an internship at a Texas worm ranch. If some of you may be familiar, they were based in Garland for a long time. And that's what got me really hooked on composting and soil biology in an applied way. But of course, it was really hard for me to find a job in this very specific niche. And so I ended up working on aquaponics farms in Austin. I eventually landed a job in plant research for a bioengineering company that specialized in lighting for greenhouses and vertical farming. So I was testing the effects that different light qualities had on plants, which was very interesting, but I was cooped up in a lab most days, and I really wanted to be on land and be using a lot of the things that I had learned through experiences, both in the lab, but also just in theory, through my education. I wanted to be applying that on landscapes. And so that's what brought me to a program called Dr. Elaine Ingham's Soil Food Web. So some of you may be familiar with that. Dr. Elaine Ingham is like the mother of soil biology. We have her 40+ year career to thank for our understanding of these microbes below ground and what they're doing for us and what they're doing for the plants. And so that's how I got the education I needed to kind of pivot my career into this more applied science. What I was finding in the career before was that I had a lot of opportunities to go to conferences, but I was just mingling with other scientists, and I rarely got to spend time with actual land stewards or growers. And so I decided that a career in research was not for me. I really wanted to be taking these ideas that research has discovered and applying them with land stewards. And so that's what I get to do today with my business, Rhizos. I serve clients of all different kinds. It's really been interesting who's found me and who's been able to make use of the microscope insights. There's sports turf managers to farmers, of course, and ecological restoration. So if the land has a soil system, then this stuff applies. And even in cases of hydroponics and aquaponics, soil biology is at play. So my intention for this session today is that I hope you leave perceiving your plants a little bit differently. And I hope that you perceive your actions a little bit differently as you engage with your gardening practice or any kind of site restoration work that you get to volunteer with. And then, of course, feeling empowered through that new perspective that's gained, so that you can steward from a place of an ecological mindset is what we're trying to cultivate today. So typically a synopsis or summary statement would come at the end, but I'd actually like to start with it to kind of frame what I'll be getting into the details of today. So what I really want to share with you today is that plants have a microbiome, and that microbiome is often compromised through modern land management techniques, but there are very practical ways that we can actually respond to common disturbances and restore the health to our soils, and therefore support the health of our vegetation. So the basics, the needs to know. I'm sure a lot of you have seen this diagram, the soil food web. This diagram is credited to Dr. Elaine Ingham herself, who provided it to you in our CS, and it's used a lot in agricultural contexts. But again, this is taking place in any soil system. So when I look at a food web, whether it's the soil food web or like a typical above ground food web, what I see is a representation of predator-prey relationships. So in an above ground food web, a classic example would be, the wolves of Yellowstone, feeding on elk and deer, right? And when that predator was wiped out once in our history, we saw the devastation that occurred from a prey population that got out of control. It took down the vegetation, which compromised habitat for a lot of other species, and overall decreased diversity across that landscape. So it had this cascading effect. And then when we saw the wolves return to Yellowstone, balance and diversity was more or less restored. So those predator-prey dynamics are actually happening in the soil as well. It's just on a microscopic level. So when I'm looking at soils through a microscope, I'm assessing who's there. What's the predator population? What's the prey population? Do we have nutrient cycling through that action of a predator consuming prey, and then their byproduct, their waste, becoming plant available nutrients? That's a huge part of what the soil food web is. And our main prey organism groups are bacteria and fungi. So a lot of times what I see in soils is that not only do we have a similar situation as what I shared about Yellowstone, where like the prey population has increased to a point of detriment to the overall ecosystem, but I'm also seeing a lack of diversity among the prey organisms. So in a lot of cases, I don't actually find much fungi. I'm only finding bacteria, and then even among that bacteria, there's not a lot of diversity. So that's a sign on the microscope that I'm looking for to kind of inform me how much work we have to do here. And the approach that Elaine Ingham and others have come to is we're essentially trying to introduce these predators to our soils and restoring that ecosystem from the ground up. And then, of course, I can evaluate for any kind of detrimental organisms, so which we typically find pathogenic organisms in low oxygen soils. So that would be called like an anaerobic soil. That comes oftentimes with compaction and flooding. We'll see an increase in pathogenic organisms, but a soil that has a thriving soil food web, even during times of flooding, will be more resilient in the long run and be able to overcome any kind of pathogen outbreak, if you will. So let's talk about the services that these microbes provide to us by way of our landscapes. One thing that we've already talked about is nutrient cycling. So again, as that predator consumes prey, they take the nitrogen essentially that they need and then they poop out what they don't. And that waste, that byproduct is actually what plant available nutrients are. And that's how we've gotten away with, that's how plants have gotten away with growing for millennia before the advent of synthetic fertilizers. There's also the service of providing pest and disease management. So this comes from a couple different modes of symbiosis. So for one, if our plants have access to all of the gamut of nutrients they could possibly need through these partnerships, then they will be able to make things like secondary metabolites, which will actually kill or repel pest and disease. But they're also, the beneficial microbes, a thriving soil food web is going to also outcompete a lot of opportunities for pest and disease to take hold in the root zone or elsewhere. So as you can see on this diagram, I love this diagram. This is a depiction of what's called a holobiont, H-O-L-O biont. And a holobiont is basically an assemblage of a host organism and all of the other organisms that coexist with it in a symbiotic relationship. So you and I are holobionts, plants are holobionts. You know, we have a gut microbiome, there's a microbiome of our skin and our mouths, and without it, we would become really sick, right? It would compromise our immune systems, and the same goes for plants. The third ecosystem service that a functional microbiome provides is soil structure, and this one really excites me. It's like one of those things that was probably a snooze fest for a lot of people in college, but soil structure is super sexy. Basically, all of these beneficial microbes are creating aggregates, and that's generating pore space for water and air to move through our soils, which is so critical, especially as we are facing more extreme weather events. If we can improve soil structure, those landscapes will be more likely to act like a sponge. So of course, when it rains, it pours, especially in North Texas, and a spongy soil structure is really necessary to help infiltrate water as opposed to allowing it to run off and go towards storm drainage. And as it is intaked through the spongy soil, that is cleaning the water. So then we have cleaner water recharging our aquifers, and we'll have the water when we need it come a time of drought. So the plants will be able to access that water directly through pore space, but also through the microbes themselves. So there's actually research that shows that fungi, for instance, which are basically, you can think of them as an elongation of the root system, this high surface area filament throughout the root zone can actually acquire mostly phosphorus, but also water in some cases for plants during times of drought. And then of course, decomposition. Can you imagine a world where nothing decomposed? That would be wild. So, you know, a forest system has a lot of leaves and wood chips falling. A prairie ecosystem has a lot of vegetation dying back on and off. And all of that needs to be decomposed and returned to the earth to maintain a really healthy ecosystem. And a functional soil food web will do that for us. So one thing to look out for in whatever context you find yourself in is looking for signs of decomposition. If you find that your mulch of choice isn't actually breaking down very readily, that's a good sign that your soils could benefit from something like a compost extract, which would inoculate it with beneficial microbes. And we'll get more to that in a second. Just one moment as I take a sip here. Okay, and then last but not least, functional microbiome will improve carbon cycling and storage. So the plants are the conduit for carbon to transit from the atmosphere to the soil. And that's called the liquid carbon pathway. It's a fun name for basically the plant taking in CO2 during photosynthesis and creating sugars. It will take a lot of those sugars and make up its own body, right? But up to 80% of those sugars are actually being released as root exudates. And the reason they do that is to serve as currency with these microbes. That's the plant's role in the symbiotic relationship, is providing that source of carbon, which then makes up the bodies of the microbes, especially fungi, and we'll take a look at fungi, but you'll see why it's the superhero of carbon storage. And as I mentioned earlier, we don't have a lot of fungi in a lot of urban soils or other highly disturbed sites. So if we can increase a lot of this bioactivity, we are effectively drawing down carbon in those soils. Okay, so just taking a pause here to kind of ask the audience, and I know I can't really interact with you all here, but maybe just take a minute and consider, what are some common disruptions that might alter, like it might interfere with this relationship that a plant has with its various microbiota? I like to think of this when I'm communicating with clients. I feel that it's my job to bring their awareness to some unexpected sources of disruption and to really tease out and investigate what might be happening in their particular context that could be disrupting the relationship that a plant has with its microbiome. This is especially the case when people have been providing a lot of efforts to try to inoculate their root zone, inoculate their plants, and we're not seeing a lot of success, or the amount of success that we would expect through that amount of efforts. We have to really tease out, okay, what are the other factors that might be harming the microbes, and we're just not aware of it yet. So that's why I've separated these common disruptions into like really obvious category and then more covert sources of disruption. So of course, like things like excavation, wildfire, floods, like any kind of devastating weather event, heavy tillage, overgrazing, all of those things, you can look at a landscape with your naked eye and not be a scientist and say, that's very disruptive. Like that's definitely going to harm this relationship that we're trying to foster. But what might be harder to interpret is when you just have, bare ground, so unmulched ground or unvegetated ground, which often goes hand in hand with compaction. Anything ending in acide, of course, pesticide, herbicide, salt-based fertilizers as well. All of those things kill. That's literally what that means. That suffix means to kill. And so they're not just killing the weeds that we're targeting. They're not only killing the specific fungi we're worried about. They're often killing a lot of these microbes. And then even if we're not using those things ourselves, we unfortunately, especially in urban contexts, but even in rural contexts, we have to be very aware of the reality of toxic runoff. And what I mean by that, or drift, right, is that a farmer nearby may be spraying one of these herbicides. And unfortunately, a lot of that carries in the wind and ends up on properties far and wide. So I want to keep perspective here, because it's not all bad news, of course. And the thing is that we're just in a point in history where our landscapes are really disjointed, right? So we live in a time where this disconnection between a plant and microbiome is really likely, actually. So it's just good to be mindful of that and consider, what it means in our context to quote unquote put the whole back, which is a phrase that I'm borrowing from a gentleman named Alan Savory of the Savory Institute and the Holistic Management International. He uses that phrase to describe what happened in a landscape when they returned predator-prey relationships between like macrofauna, like large animals, and how everything kind of bounced back on that landscape to become more functional. So what it might look like to put the whole back in our little parcel of land that we have any kind of access to or effect over is by being mindful of the plant microbe relationship and putting the whole back, we will effectively increase water holding capacity, will affect above ground biodiversity, and will affect the resilience of that landscape. And I'm sure a lot of you actually have examples of this, like when you've planted a garden and you've seen the effect that it's had on the types of birds that visit, or the type of lizards and frogs and so on and so on. It is like a call to the surrounding area for more life. And this can start from the microbiome below ground as well. Okay, so now we're gonna have some fun. We're gonna look at some examples of microbes that I get to look at on the microscope. So the first up is fungi. And so these long branching filaments are the hyphae. So in a textbook, you'd probably hear about mycelium. And so hyphae, I think of mycelium as a collection of cables, and hypha or hyphae being just one of those cables within the conduit, right? So that's what we're looking at here. So the video is a image of fungi at 400 times magnification. And you might notice, depending on how close you can see this video, something swimming around in there that is a flagellate. So that's actually one of the predators that will consume bacteria and make nutrients available to plants. This looks really similar to what you'd probably see fungi depicted as in a textbook. It has the strong hyphae walls and the septate, which are the providing that rigidity in between. And this is why fungi is like a carbon superhero of the underground, is because that rigidity, that structure, is all carbon based. So a carbon atom is a very stable atom and able to build these really complex structures and really rigid, stable structures. So that, if you can imagine the amount of surface area you get out of this thin of a filament going all through a root zone, that's how we get carbon to be stored long term. Where the carbon cycle among these bacteria and protozoa are more short term, still very valuable, of course, to the whole ecosystem. But in terms of carbon drawdown, if that interests you, fungi are definitely the organism group to pay attention to. Okay, here's a more charismatic character. So this is a bacterial feeding nematode. So if you see, it has some, we would call these fancy lips. And that is meant to just slurp up all those little bacteria. So the bacteria, by the way, are these vibrating specks that you see around. And so while this nematode isn't actively feeding, we get a really clear look at its digestive system and this outer cuticle. And of course, those fancy lips. So I see a little bit of fungi off to the right here. These dark bits just kind of scattered about, that's organic matter. So that's actually like little micro aggregates. Again, this is at 400 times magnification. Okay, This one's kind of neat. You'd have to look really closely, but this is actually a little baby nematode that hasn't hatched yet. So it's just kind of moving around in its egg, probably going to come out within the day that I looked at it. Again, a little bit of fungi here, organic matter. We see bacteria scattered about. So the bacteria are just kind of vibrating with life. Some of them are motile and will actually swim around, but here we just kind of see them moving ever so slightly with life. Okay, everyone loves a good naked amoeba. Naked amoeba are what you think of when you think of an amoeba. It's an oozing blob. So here in the center of the field of view, it's kind of got this funny shape, and you'll see as it moves, basically sensing which direction to move in to feed and get where it wants to go. So it just has a really sensitive way of moving and the ability to do so that other organisms don't. So that's pretty cool. And on a lot of naked amoeba, you can actually see what's called a vacuole on it. And this one, I think, is kind of in the center here. I hope y'all can see my cursor. I'm not sure if you can, but the vacuole is collecting the waste, and eventually it'll flush and release all of those nutrients to the root zone. This here is another bit of fungi. It's less fun when I can't hear all the reactions in the room, so I hope you're oohing and aahing. But this one's really cool. This is really rare. So a lot of amoebae will actually be in a test, like a little shell, and that makes them hardier. I see these a lot here in Texas, given like our heat and stuff. And I see these a lot in hot compost. So where I rarely see naked amoeba, I usually see naked amoeba from like samples that are sent from cooler places or in cooler seasons here. But anyway, the test, yeah, it just helps protect the amoeba. And so when I look at it on the microscope, a lot of times they'll just be tucked inside their little shell and scared and not want to come out because I just shook them up in a test tube, right? So this is a rare occasion where I actually saw it come out of its shell, and it had long tendrils. So it's pretty differently shaped than the blob that we saw before. And it's just moving right through the soil media, which is pretty cool. I'm pretty sure this is the tail end of a nematode off to the right. Oops, didn't mean to. Okay. All right, it gets cooler. So my next one is my favorite. It's a flagellate. And this is the coolest flagellate I've ever seen. So it's, I'm showing you my highlight reel, of course, but it starts out as this weird mushroom shape and kind of morphs into different shapes. And that long, if you can see it, there's like a long whipping tail on it. That's the flagellum that propels it and makes it motile. So it'll actually kind of start dragging itself here through this sample. And again, flagellate and amoebae, they're predators of bacteria. So they're providing all that good nutrient cycling. Yeah, watch this. It kind of elongates out, and then it's no longer this like mushroom shaped thing. I've only seen this type once since then. It's been from the same client's soil, so pretty exciting. Most flagellates are like a tenth of this size. So this one I posted on my Instagram to elevator music, which was fun. It's taking a little while for the slides to load just because of the amount of video, I imagine. So I'm just going to be patient here. Okay. All right, so that's the end of our highlight reel, but here's something important I want to convey. So on the left, we have an example of what dysfunctional soil looks like. So I say dysfunctional because it's basically like I'm not seeing all of these characters in the soil. So we know that it's not operating at an optimal state that would be providing all of those services to our plants and landscapes. But on the right is an example of a very functional soil that has a lot of biodiversity and providing a lot of those ecosystem services. So one thing to note before I play these is that the one on the left is 4 times more diluted than the one on the right. And I share that because of the amount of bacteria that you'll see in the one on the left compared to the right. And just know that it's been diluted. So here we see, this is a nematode kind of flapping about. We see aggregates. We see these little micro aggregates, organic matter. There's bits that there is a testate amoeba. There's fungal spores, a little bit of fungi here and there. So this is incredible to see this much diversity in one field of view. Again, we're magnified 400 times, and I'm just looking at a single drop of a soil sample. So it's pretty incredible to see that amount of biodiversity in one field of view. And then here on the left, all of that there that you see is bacteria. And what I'm doing here is I'm focusing from the top of the drop down, because you don't think of it, but a drop under a cover slip actually has a depth to it. So we're focusing through the depth of that. And there's a few bits of aggregates here and there, a little bit of like mineral matter, those kind of glassy looking bits. This is probably just mineral matter, but I'm not seeing any other characters from the soil food web. And when I look at a sample like this, I'll look at 50 fields of view. That means I'm moving the microscope around the slide 50 different times to get a sense of the whole sample. And all of them might look like that. And I won't come across any a nematode or amoeba or a flagellate at all. So this just helps you kind of depict how I view, so the soil food web under the microscope. Now, one thing I want to share. Oh, I see what's happening now. I'm actuating it on 2 different screens, so that's probably why it's delaying. Okay, so now the important part of this puzzle, and I didn't know this until I started this work and using a microscope, but I started looking at compost as well, of course. And again, on the left, we have a compost that I got from an industrial source, like a municipality. And then on the right, I have a biodiverse compost that was made at home. So I'm playing the videos now. The one on the left just started. I'm playing the one on the right. So again, we see a lot of bacteria up here on the left. There's a flagellate swimming through. This is fungi right here. We see a testate amoeba at the bottom, a little fungal spore, some aggregates. This is actinobacteria, which is something we hadn't talked about just yet. And if it didn't already, I think there's a nematode that photobombs that video too. But anyway, you get the point. It's a very similar story between dysfunctional soil and the difference between industrial compost and a really biodiverse compost. And so this begs the question, why are we applying bacteria dominant, low biodiversity compost onto disturbed soils that are suffering from that very thing? So a lot of people don't know this, and this is like new information, right? A lot of us go and get compost year after year, and you use it thinking that we're providing a huge service to our plants. And I'm not suggesting that it's useless, but from an inoculation standpoint and repairing the soil microbiome, it's really not useful. In fact, it can actually make things worse from a biology perspective. I liken this to if you had a mouse problem and your solution was to go to the pet store and get a bunch of mice and then release them into your home. Like, it doesn't make any sense. You're just adding more of the thing that like you really don't want more of. Okay, so now getting to some practical, what do we do with that information? The big thing here is improving the quality of the compost that we're using. If you're not using compost in your various contexts, I'd encourage you to start using some high quality compost. And so the question is, how do you know what's high quality compost? What's a good biological quality compost? One thing that I tend to get philosophical about is a lot of compost makers don't have any reason to be making better quality compost because the consumers aren't aware of the difference in biological quality between the stuff in a bag at a big box store compared to something that like Texas Worm Ranch was making in Garland. And I say was because they've moved to New Mexico, so they're still around. But the biggest thing that I do in my community is teach people how to make excellent compost. And I like to do it in one of two ways. Well, actually one of three ways lately. So I have depicted here something that's called a Johnson-Su bioreactor. So if you've not heard of that composting method, I just want to introduce you to it here. It's got a fancy name, but it's not going to take us to space. It's just a cylinder of compost. And so, and I can drop it in the chat so you get the spelling right, but it's two last names. It's a husband and wife duo that created this method, Johnson and Su, S-U. And then the term bioreactor is what they've dubbed this system. And this is basically like an incubator for fungi. So you fill it up with exclusively fungal foods, like really carbon-based input, so wood chips, brown leaves. Basically, in the fall, when everyone's throwing their leaves out, you can just take those and fill up a cylinder with it and grow a lot of fungi. The principles I go into in a few different areas, which I'm happy to share, but the big thing is keeping it moist and making sure that has good airflow. Those are the two things that fungi really need to increase in number in this composting pile. But what I love about it is that it's super simple. You build it and then you just maintain moisture and set it and leave it. You don't have to turn it. It doesn't get hot. Like, it's very easy. And the only downside is that it takes a while. So you'd come back in 9 to 12 months and see if it's broken down to a nice like clay-like consistency. And this stuff will just dissolve in water and you can start using it in your gardens to inoculate. It'll be the best inoculant you can make. You can't even buy inoculant better than you can make at home in a Johnson-Su bioreactor. And then the other method is vermicomposting. This is a very poor example of that. I think this is actually worms from a soil sample actually. But anyway, you know what vermicomposting is. And vermicompost is, it can go wrong, okay? But for the most part, the worm castings are going to be pretty biodiverse. One thing that you can do to improve the microbiome of worm castings is to actually feed them a little bit of a fungal food. So the easiest way I find to do this is like a little bit of oatmeal or oat flour can help boost the fungal biomass in that finished compost significantly. So the tendency for vermicompost is for it to also become a bit bacterial. It'll almost always have protozoa and nematodes in it, but just not a lot of fungi. And most of our landscapes would like some more fungi. So one way to help improve the quality of your worm castings is to make sure that you're adding a carbon to the system. And that happens, by the way, anytime we're composting and we're just doing a lot of nitrogen-based stuff into the compost, that's bacteria that are going to take up that nitrogen, and the fungi really need a carbon source. So that brings me to my third composting method, which isn't depicted here, but just like a good old backyard compost heap. And you know, you could go cross-eyed looking at all of the recommendations online for how to compost well in your backyard, but I like to keep it really simple and just tell people the best thing you can do is try to keep a 50 to 50% ratio between your carbon inputs and nitrogen inputs. So for me, what that looks like is if I take a bucket's worth of food scraps to my compost pile, I'm going to make sure that I have a bucket's worth of leaves or wood chips and or shredded paper, something that's like carbon. So I also have gotten in the habit of just keeping a pile of wood chips and leaves near my compost bin, because I'm often bringing the stuff that I'm bringing from the kitchen is almost always going to be nitrogen based, like food scraps, just consider them nitrogen. Coffee grounds is very nitrogen rich. So anything that's coming from my kitchen is, I've already got the nitrogens covered. What a lot of people lack is, where do I get my carbons from? So if you can set up a system with your wood chips or carbon source nearby, that'll ensure that you are maintaining that balance. And then, yeah, we'll use that compost in a number of ways. So I like to make extracts because they're very easy to make in just like a mesh bag in a 5-gallon bucket. You put your compost in there. All we're trying to do, we're not doing a tea, we're not like brewing it, and we're not getting too fancy. All we're trying to do is just knock the microbes off of the substrate of the compost and into the water. And then we use that water with a watering can to, just in small scale applications. You can also use it as a foliar spray. Going back to that image of the holobiont where you have the microbiome all over the plant, you can also spray foliarly, especially if you're seeing signs of disease. Go ahead and spray the plant parts. And then, of course, you can use this for seeds. You can just you don't need to soak them too long, because that tends to make them swell and germinate maybe sooner than you want. But you can spray seeds with an extract, or maybe just dunk them and then kind of let them dry and then use them to plant. Okay, and then mulch, right? So really thinking about the context of native plants. I'm working on establishing a pocket prairie right now. And I've been considering, how do I want to mulch that long term, because it takes a long time for native seed to germinate and get established. And so I went out and I bought native grasses so that I'd have some kind of seed stock, if you will, to mulch the area with something that nature would have already mulched itself with, right? So taking shears at different times of the year to lay some of that leaf litter down, some of that grass down, to protect the soil, to make sure it's never bare. You can also do this with leaf mulch or kind of a finer wood mulch. The big thing with mulch, if you're going to buy it, is that I prefer using an aged mulch because a lot of mulches are meant to repel breakdown and repel insects, and they will eventually degrade, but it's just easier if you have a cedar. No, there's nothing wrong with getting a cedar mulch. It's just if it's aged, it'll be easier to use on site because it'll actually start degrading faster, which is really what we want. It might even feel counterintuitive because a lot of gardeners think, oh, I don't want to have to re-up my mulch later this year. But we really do want that stuff breaking down. That we want an organic mulch layer that's actually feeding the soil and protecting the soil surface. And then lastly, pruning. So this is going to look different depending on the context, but this actually will stimulate root exudates as the plant kind of signals. It is mimicking grazing, essentially, when you prune a plant, and it will generate more root exudates, which will improve that relationship between the plant and the microbes. On that note, I did write a whole post on soil regeneration without livestock. Like basically, how are we going to take the role of the bison? If you go back to that slide where the image of all of those farmscapes and the suburban landscape, it's all cut up, and like we could never graze that like it once was, and restore the ecosystem in that way. So, but we can consider like what was it that the bison were doing to generate that kind of vibrant native system and take on some of those responsibilities. And that, the last slide being they inoculated with their gut, right? They introduced organic matter and they disturbed the landscape a little bit. So aeration was something we didn't talk about, but that could be incorporated. But this post really goes into each one of those. And I actually had a reader turn it into an infographic, which he printed and put in his office in Pacific Northwest. Like he is a subscriber and he owns a landscaping business. And so he put this up in their office. I thought that was pretty cool. You can actually download this infographic and print it yourself on my blog, which is called Soil is Sexy. A fun, playful, free place on the internet. And then another resource is, which we didn't get into at all today, is a deep dive on biochar. So what's cool about biochar, like in the way that I'm using it in my pocket prairie, is native landscapes would have naturally had biochar from grassland fires, and I'm trying to establish one basically on subsoil because of excavation. And so it just seemed like a cool thing to incorporate biochar into the soil along with compost to help these plants that I'm seeding. And then lastly, I have a post that's super straight to the point, how to make a compost extract at home. And that one's called Compost Extracts 101. So if any of that interests you, here's a QR code for my blog. And then of course my business provides lab services if you're interested in checking that out. But I am ready for your questions. I hope I was honest on my timeline. Okay, I'm looking at the chat for questions. I hope that's the right place.
I tried thing with worms, but I found in the heat of Texas, they don't survive in the bed. How do you do it in the heat of Texas?
Okay, her question was she's tried worm composting here in Texas, but her worms wouldn't survive because it was so hot. So how can you do that in Texas?
Yeah, that is a challenge. So I've always had mine indoors and they do well. I have seen people be successful outdoors in at commercial scale too, like small scale and commercial scale. And their trick was to make sure that I was in a lot of shade, right? So like almost constant shade, and a very deep leaf litter. So in one instance, somebody had them in like this old recycled bathtub, and just maybe like 4 to 6 inches of brown leaves were on top, and the bedding for the worms was below that, and it was under a large oak tree, and it would survive every Texas summer that I got to see it. So yeah, it is tough. I do want to acknowledge that, but either bring them indoors, like keep your container small enough that you can bring it inside, or try to create some kind of shade structure for it and do deep bedding and see if that gets you anywhere.
How quickly would you say that they can move through healthy soil in like a day's time? Can you repeat the question?
Her question was beneficial nematodes. How far can they move through the soil in a day?
I don't really know. I don't know. I would ask you, I'm curious, like what's the interest driving that question? Are you trying to get nematodes to arrive somewhere in a certain time? Like if you know that you have good healthy soil nearby and like you're wondering maybe like if they'll move and become present elsewhere, that would, I would say, is dependent on conditions, but I don't know at what rate they can really swim. I will tell you that in my lab days, we used to buy beneficial nematodes in like a little pack, and they would, if you left the pack unattended for 5 minutes or less, they'd be like out and like on the lab bench. So I mean, they can move faster than we probably give them credit for.
So I have ants that they're not fire ants, and I don't know what kind of ants they are, and think they might be the invasive crazy ants. If you've heard of those? But they're in various different places in my backyard. And I did buy some beneficial nematodes, but I was just curious as to how quick they're gonna work for you.
Okay.
So, like how far and wide those nematodes can go to do the job.
So she has ants in her yard, and she doesn't think they're native ants. They're not fire ants. She thinks they're those crazy ants. And so she bought some nematodes, and she was wondering if she put those out, how far and wide those would work to, I guess, maybe deter the ants, take care of the ants.
Yeah.
Eggs of the ants. So
Eat the ants' eggs.
Yeah. I mean, if you're able to identify where the ant's origin is, like their nest or something, then I would expect the nematodes to be pretty quickly effective to reducing their population. However, the only concern there is a lot of these nematodes that we can purchase online might not necessarily survive the conditions that we have in our particular context. So that's always a risk, and that's another reason to get into composting, because you know that the microbes you're raising right there are going to be able to survive your particular conditions when you use them as an inoculant in nearby soil.
What is the proper percentage of
What are the proper percentages of biochar?
In the soil.
So in that post, I interviewed a biochar expert, somebody who studied it for their master's degree, and it's very well sourced in the rationale that he presented in that interview. So I highly, highly recommend you read that, because at the end we talk about kind of the do's and don'ts of biochar, but a lot of the decision making around how to use it goes back to context, which is, one of the soil health principles. So to answer your question as simply as I can, I personally, after meeting with that expert, I err on the side of less biochar than somebody selling it might recommend. In cases where I know the pH to be rather alkaline, I'm particularly careful. So I keep it 10% or less in alkaline soils and up to 20% in other contexts.
What was the source of that study?
So where you can find that information is on the Soil is Sexy Substack, and there's an article called Biochar, What to Know.
So it's on her website.
Yeah, and this will be the image that you see there. And we have like a long list of references where all of that information was sourced for that post.
All right, my question is, how often do you apply compost during the growing season?
How often do I apply compost during the growing season? So I am of the mind that you can't really overdo compost extracts, and that is my preferred method of applying compost. If I were to be establishing a garden for the first time, I might use some solid compost to mix into the topsoil and top dress a little bit, and then mulch on top of that to protect the microbes that are in that compost. So that's one thing to just be mindful of if you're using compost as a top dressing. But for compost extracts, I'm a kind of a lazy gardener. I mean, that's why I grow natives and not like a bunch of vegetables. So I do it when I feel like it. And it probably is like during the growing season, I might do it a couple of times at the start of the growing season, like a couple of times within the first week of transplanting something or seeding, and then maybe like once a month from then on.
Also, what if they are potted plants? If they are potted plants, would you do it more often?
Yeah, would I use compost extracts more often for potted plants? That's a great question. I mean, the thing with a potted plant is that it doesn't have access to that large of an area. And so I do think that inoculating potted plants more regularly would be beneficial to them. So yeah, it's up to your bandwidth, but if you have excellent compost, just use it. And a little bit goes a long way. Like that Johnson Sioux bioreactor, all you need is a little bit to make a large batch of extract. So I'm talking like a cup's worth for a 5-gallon bucket that technically you could spread across an acre if you had the equipment, but like we're just going to be using it probably by a watering can with individual plants or small garden areas. So that Johnson Sioux should last a long time for smaller contexts.
Would compost tea be better than actual just compost?
Is what you're calling compost extract, is that compost tea? And she is asking, is that better?
So the difference between compost tea and compost extract is that tea, at least, and this is like among professionals, so like you might be using it to mean the same thing, which is fine, but compost tea technically means that we're adding microbial foods and we're brewing the microbes. We're trying to increase their numbers in a lot of cases, but I don't recommend that for hobbyists because what inadvertently happens is that a lot of people just end up breeding a lot more bacteria. And as we just talked about, like that's not really what we want to do. So an extract is usually more foolproof, and it's also just a heck of a lot easier. Teas will also vary significantly depending on the temperature outside. So like when I make teas at home during the summer, I have like an 8-hour brew period compared to like a 48-hour brew period in the winter. And all of that's really technical and nuanced, and it's tedious. So I would recommend extracts. And then remind me the second part of the question, like, do I ever just use straight up like solid compost?
Are you asking if the extract is tea is better than extract, or solid versus an extract? Which
I think I like extracts over solid compost only because it's kind of like a carrier for the microbes to soak into the ground. So the best practice for extracts is to wet the ground first with water, or maybe after a rainstorm or something, and then come in and apply your extract, and it'll actually soak in the ground to get the microbes where we want it, as opposed to top dressing, which tends to just kind of sit on top. I also have, I can send y'all some links too if you want to send it to your email listserv. I can send you links to the biochar. I've done some write-ups on Johnson Sioux and others that might be relevant to our conversation. How to make compost extracts. Yeah,
that would be great.
How do we do a biological test of the soil? Is that right?
Yes.
Yeah. Now we can
Yeah.
Yeah.
Yeah. So I mean, I'm biased, right? But I think the best way to evaluate the biology of a soil sample is with the microscope. So I just want to acknowledge the other methods that, like, if you're used to going to a certain lab for your soil chemistry testing, they might have something called PLFA. That stands for phospholipid fatty acid test, and that's like a marker of biology. it's a very indirect measurement, but it will at least be relative to you over time. Like you'll be able to see a trend of if things are becoming more diverse or more abundant. Whereas microscopy is a more direct observation because it's as close as we can get to like putting our eyeballs underground. And it comes with its limitations. You know, we're only able to look at a drop of a sample of a large ecosystem, right? But the Soil Food Web has a website with certified lab techs like myself, and you can find one in your area. I'm starting to, one thing I will say is that the data collection for the microscope work can be pretty expensive. So most of my business is business to business, and because they're $120 per assessment, which comes with a discussion document and a follow-up call and things to help large producers improve their soil health. However, I'm going to be I've started providing qualitative tests, which are, they don't result in data, but it'll at least provide insights into your microbiome descriptively and come with photos and videos of the observations we saw under the microscope. So if that's helpful, that's $40 per sample.
Jim asked, why is commercial compost poor in quality?
Why is commercial compost poor in quality?
Yes.
Okay. There are a few reasons. One is actually their business model. So the way these companies make money is primarily through the dump fees. And so they're motivated to grind that up and make it look compost-like, soil-like, and get it out the door as quickly as possible. They're not really in the business of making a very useful soil amendment. Maybe they would if consumers were putting pressure on them. We'll see how that plays out. So one thing that I do encourage people to do is to call and ask, just ask a general question like, do you have any biological, do you know the biological quality of your compost product? Or what's your fungi to bacteria ratio of your product? These are things that a very thoughtful compost maker will likely know about. And this isn't supposed to be like a gotcha or like villainizing anyone. It's just to let people know that we're interested in the biological quality of the product so that they can know that there's demand for that. But the physical reasons that the compost doesn't end up being very, biodiverse, and this goes for every compost. The resulting microbiome depends on the feedstocks that are going into it. So all of the inputs, the recipe, you kind of need a balance of carbon and nitrogen to have a balance of fungi and bacteria in the end. And you can't really do that in these commercial composting facilities because, again, they're just dumping off a bunch of brush and they're grinding it up. And my colleagues and I actually call it GOM, ground up organic matter, to differentiate it from actual compost, like useful compost. So that's a term that you can take with you. Am I using GOM or am I using compost? So the feedstocks and the moisture and the heat. So sometimes their only regulations are to get to a certain temperature to make sure that there's no human pathogens in it. And while I appreciate that, there's like no other regulations for biological quality. So what tends to happen is that the compost piles get too hot for too long and they just kill everything and they don't give them enough like rest time to mature. But I've also seen where even if there's a maturation phase, the microbiome never rebounds because the feedstocks are gone, the nutrients are gone, so there's nothing for them to really feed on and improve the overall populations. And then a lot of times they're underwatered. Just like you and I, if we were underwatered, we would come out a little haggard ourselves. So hope that answers your question. But yeah, it's complex. And I really think all this could change if their business model was a little bit different, if they were really in the business of making soil amendments rather than compost, which is such an ambiguous term that we use in so many different ways.
Is commercial mushroom compost better than other commercial compost for overall soil health?
Commercial mushroom compost, is that better than other compost? You know, I haven't looked at many mushroom composts. I've looked at like leaf mold compost, and I was disappointed. So I'll say that. But I do like using mushroom substrate from you know, mushroom, like culinary mushroom growers will use their substrate in composting. And I do think it's a good fungal food source. But yeah, I would just go back to the principle of asking the maker, do you know the biological quality of this compost? And what I'm hoping that they would respond with is not sending you their E. coli reports, which is what the regulation is about, but sending you something more like a soil food web report that emphasizes the diversity in it.
Can you retrofit commercial compost by introducing microorganisms, not bacteria, such as soil additive compost seeds? So can you take like commercial compost and like zhuzh it up?
Zhuzh it up. I love this question. That's like exactly the way that I want everyone to be thinking about soils and like the resources that we have access to. So a lot of my clients, especially in like small organic farming where they're kind of addicted to GOM, like that's just part of their infrastructure. The next best step that I can teach them is to make a small amount of really biodiverse compost and then use that to inoculate their GOM. And that can work really well. The other great thing that I've seen that's worth mentioning is some large scale compost producers are becoming more hip to this. And what they tend to do, they're still grinding up all of that debris, the brush that comes in from the landscaping crews, but they'll actually create like a slurry of other microbial foods to then spray on that. And that tends to have the effect of improving the microbiome. It's still not going to be as good as what you could make at home, but it's going to be a heck of a lot better than the other GOM producer.
Suzanne wants to know if predators are lost from your soil, how do they recolonize when you start working on improving the system? So I think she's saying she's going to work on it, but in the meantime, what can she do to help with beneficial predators while she recolonizes her soil microbiome?
Yeah, this is kind of like a chicken and egg question, because in theory, you know, our soils could improve on a geological timescale, but like we will be long gone before we like see the benefits of that. So that's where like our role as stewards comes into play. And I've often pondered this, like if the prey population is there, like they have an unlimited food source, like where are the predators? And I think the limiting factor for them is habitat. So going back to soil structure, like they need pore space. These larger microbes, they actually need spaces to hide and find their food and feel safe in the soil environment. And that really requires pore space with water and air throughout. So you kind of like need the microbes to generate that soil structure, but the beneficial microbes need the soil structure to survive there. So this is where we just really have to think, like, how did nature do that? And it was through, going back to that infographic, it was through these different ecosystem services that grazers provided to the landscape. and that of course is just an example of the prairie, which is what I'm focusing on because we're talking about native plants. We need to be kind of like moving the ball forward on a couple different planes. So could we have a really useful effect just inoculating alone? I think so. I think that's the most important one. But we could also be moving the ball forward by increasing organic matter and doing maybe a little light aeration and disturbance and pruning and all of the things that bison would have been doing on that landscape.
Would like to know, what should she add apart from dry leaves and green grass and fruit vegetable scraps or threaded newspaper? Like, there's something specific?
To the compost, I imagine.
Yes.
Yeah. Just going back to that ratio. So something to keep in mind is that, if you freshly clip grass or pull weeds, those as they're fresh are green. They're going to have a lot of nitrogen in them. But if they sit in a heap and are exposed to the elements like the sun and the rain and the wind, eventually they'll brown, and then they become more carbon based. So, and they're not a particularly, I wouldn't rely on that as your exclusive carbon source. I really think that a more complex carbon source like wood chips is really essential to getting a balanced microbiome. But it's not to say that you can't use dried greens as a source of browns on occasion. The key is diversity, which it sounds like you already have a lot of in the list that you provided there. The only thing I would have you consider is kind of categorizing by volume. How much of those would you classify as greens? How much would you classify as carbons? And do you have diversity among both your carbons and your nitrogen? Sorry to be using greens and nitrogen interchangeably, but I try to stick to nitrogen and carbon because that's like very literal. When like you say browns and greens, it can get confusing because something like coffee grounds are like brown, but they behave like a nitrogen. So I try to stick to nitrogen and carbon.
Me too. All right, Grace, can you recycle the compost back to the pile after dumping it in extract?
I would use it in a new pile. So to repeat the question for the people on the webinar who might not be able to hear that, can you, like after using compost for extract, can you then recycle that compost and put it back in the compost bin? And my suggestion is that I would use it as a source of carbon material for a new pile. So that's why a lot of people get in the habit of having kind of like one finished pile that's being stored and you're not actively adding anything to it. And then you have one that's actively receiving feedstocks. So that's what I would do with that. And the reason is just that at that point, that's what it's most useful for, because you've taken the microbes off of it. It won't really break down too much more than what you've already done with those microbes. So I think it can be a useful pseudo inoculant too. Like it's going to have a little bit of life in it, and you can inoculate an existing kind of active pile with that, and it'll become a carbon source for that newer, younger pile.
Right. Jim wants to know how you feel about hydroponic vis-a-vis the import soil.
I'm sorry, I might have missed that one. How do you feel about what?
Hydroponics.
Hydroponics.
The importance of soil. So again, is still in a day and age when we can grow plants without soil?
Yeah. Well, the great irony was that I was so jazzed about soil in college, and my first job was on an aquaponics farm. And then from there, I ended up going into research, which is all a sterile environment, very hydroponic based systems. And the thing about that is biology is everywhere, and it's just whether you acknowledge it or not, and whether you leverage it or not. So what we're doing in soil-based systems is really leveraging biological interactions. And in hydroponics, technically you can, but gosh, you got to really orchestrate it, right? I mean, the organisms can live in a properly aerated hydroponic system and cycle nutrients and all the things. But you, sorry, I got a little feedback, which threw my concentration through a loop. They'll technically survive in a hydroponic system, but you have to then be the source of the nutrients for them. Does that make sense? So it just kind of complicates our job as growers. I will say, I think hydroponic systems can be useful, and I think they can be fun, and I think those are good reasons to explore them and enjoy them. And you can still use soil food web principles as you work with a hydroponic system, especially an aquaponic system, which has an organic nutrient source. But it changes your perspective. So like a hydroponic grower that's used to feeding their plants directly, like plant available nutrients, who then wants to actually have beneficial biology survive, they're going to have to start feeding more things like kelp, fish hydrolysate, molasses, maybe like microbial foods that will sustain the system. Thank you. Thanks for having me.